Hi, girls. It's our second day of taking video notes. Last class, you should have taken notes on what DNA was, what it shaped like, what the structure was, and how it held your genetic information, and also who helped discover that. And many different scientists were working together to discover what DNA was, the information that it held, its importance, and what it looked like. So, today, and I'll repeat the same instructions I did with the last video. Make sure that you have your OneNote notes open to chapter 13 notes. You're going to scroll down until you see section 2, Replication of DNA. And then if you're taking notes in your paper notebook, you want to make sure that you have that ready to go also. And remember that throughout this process, you can pause the slide or pause the video and take the notes first, or either listen to me talk and then write down the information, whatever works best for you. Okay. So today we're going to talk about the very important process of how DNA copies itself. You've been hearing that since cells in January. You heard that during the interphase of mitosis, DNA had to make a copy of itself. You even heard that there were some checkpoints where some enzymes came through and checked the DNA had copied itself correctly. And if not, there was, enzymes that went back and fixed it, if possible. So this section in your book is pages 300 through 303, and it will talk about the actual why DNA has to copy itself, when it has to copy itself, and then the 3 steps it uses to do so. I'm going to go ahead and go to the first slide. So the learning objectives for this section are to be able to explain how DNA replicates itself, and then also to explain the roles of the proteins that are involved when DNA replicates itself. Okay. Move to the first slide. Usually on this slide, we're taking this in your notebook and a title that is DNA replication. How does DNA copy itself? So before cells can divide, and that's talking about either mitosis or meiosis, you know DNA must copy itself. So each new cell is able to contain its exact copy of the original cell's DNA. In turn, DNA replication, the definition is just the process where DNA makes a copy of itself. I don't know if this video will let me play. No, the links don't work. Okay, so we'll watch this when I get back. All right, so let's go on to the next slide. You only have 3 slides. It's very short today. Okay. So why does DNA need to replicate? Well, we know anytime a cell divides, DNA has to replicate. So that ties into the first bullet point. Cells divide for an organism to grow or reproduce. Every new cell needs to copy the DNA, aka instructions to know how to be a cell. Okay, so we know DNA is replicated itself right before a cell will divide, either in meiosis or mitosis. That happens in interphase during S phase. Remember, synthesis is a phase of interphase. And a really important reason that DNA has to replicate is to make proteins. We are protein-driven organisms. I'm not talking about pizza chicken. You eat, even though it's important to eat proteins because you need proteins to help your body make its proteins that keep us functioning. Right, so I want you to look at the picture that's below, and I'm going to see what color I will get. How about yellow ink? So if you look down below, you're going to see they have DNA kind of looking like a ladder. It's not spiral like you've seen it before, but you could recognize that it's 2-stranded. Here's one strand, and here's the other. And we know that the backbone is made of alternating phosphate and sugar molecules, and we know that the sugar molecule is attached to a base. And those 2 things make a nucleotide, and we know that the bases are in the center, and A pairs with T, and C pairs with G. Those are our complementary base rules, right? We learned that last class. So DNA, when it replicates, the two strands that are attached of the original DNA actually unzip because this enzyme called helicase comes up and it breaks bonds that hold the nitrogenous bases together. It breaks them apart. So it comes up through here. They call that unzipping. And so now you have one half, each half is exposed. And so each old half has new bases added to it. It's pretty ingenious. So this is our old half that is kind of a purplish blue. And this is the other old half that's been opened up. So you see new bases are added in green. It doesn't really give you letters of the bases that were added or what the code was, but it gives you a general idea and shows you. So start with one DNA strand. That DNA gets unzipped. An enzyme unzips it, and another enzyme brings the new bases over and pairs it up. So at the end, you have two identical DNA strands. Half of it is the old one because it kind of works template. It was the pattern for the new one, for the new bases to attach to. You end up with these two brand new strands of DNA. Each containing half old and half new, because where it flips open, that one half is kind of part old and part new. But they're identical, okay, because basically it's used as a pattern. It uses itself as a pattern. Very smart. So let's go to the next slide. Okay, so here are 3 steps of replication. Actually, I just realized I was sideways. I have a computer, so I was sorry. 3 steps for how DNA replication happens. First of all, I'll say unwind and unzip. That's step 1. Step 2, I'm all about the base. You know that Megan Trainor song. I never really liked the song, but it's a little great for this. And the third, twins. So what you end up with is what you started with. Instead of having one, you now have 2. Okay, because remember, it's making a copy of itself. So let's start with the unwind and unzip. DNA is unzipped between base pairs by an enzyme called helicase. The helicase breaks apart the hydrogen bonds between the nitrogen bases and opens DNA up, exposing each half. Okay? Again, DNA is open. You can look at this picture to the right to see that. See it's open, you have half old and half new. Then, after DNA is unzipped, an enzyme called DNA polymerase is going to come through there, and it's going to add the correct complementary nucleotides to each strand where it's been unzipped. So if where it's been unzipped, there's just A, what are you going to add to that A? What pairs with A? Oh, you said T, right, T. And if it's the next base is a D, what pairs with that D? C, you're right. C, yes. Or if it's a C, what pairs with that C? D. Or if it's a T, what pairs with the T? A. Good job. So DNA polymerase, it works to come through and pair up all of those bases with ones that have been opened up to get a double strand again. And then in the end, after all the complementary bases have been added to the old strand, so it's half old and half new, you have twins. Yay, congratulations, twins. So 2 identical DNA molecules were formed that were identical to the original. So 1 becomes 2. Okay? All right. And I think this is the last slide, but I'm moving to the next one. But if it is, you can go back through these notes and rewatch them.